So this is our third video for Chapter 24, Transition Metals. And this is the last video for Chapter 24, and we'll be looking at crystal field theory and how that explains the colors and the magnetism of transition metal complexes and the applications that make transition metal complexes unique and very interesting. Now, if you'll remember back to Chem 1A, and we were looking at the different bonding theories, one of those was valence bond theory. And in the valence bond theory, we had hybridization of orbitals. So we had sp and sp3 and dsp2 and the dsp2 hybridization to explain some of the bonding. And if we had sp2 hybridization, you hybridize an s and a p orbital to create a linear geometry. And for a tetrahedral geometry, you're hybridizing an s orbital and 3 p orbitals. And for square planar, it was using a d and an sp2, and octahedral, a d, 2 of the d orbitals and s, and then 3 p orbitals to create this octahedral geometries. So the bonding takes place when the filled atomic orbital on the ligand overlaps with an empty atomic orbital on the metal ion. And this explains the geometries very well, but it doesn't explain color or magnetic properties. So valence bond theory wasn't very useful to the inorganic chemists for explaining the bonding situation that was happening, and also why we saw the different colors and the different magnetic properties. We needed something new to describe what's going on, and this is where we have the advent of crystal field theory. So we needed a simple way of explaining the reactivities and the properties of transition metals. And knowing the shapes and the orientations of the d orbitals is crucial for your understanding. So remember, each of the d orbitals has the 4 lobes, where the dx squared minus y squared lies on the xy axis, and the yx lies between the zx, the yz lies between the yz, and the dxy lies in between the xy axis. And then the dz squared, we said, remember, that's the weird one that has the 2 lobes that are in the same phase, and then the donut around it. So knowing these shapes and how they're oriented will help us understand why the transition metals are different. So the bonding and coordination compounds is explained using crystal field theory. And the bonds form due to the attraction of electrons on the ligand for the charge on the metal cation. So remember, again, think about our Lewis acid-base chemistry. So the ligands have lone pairs that can donate electron density to the metal. So the metal acts as an acid, and it forms this coordination bond. So it forms this Lewis acid-base adduct. But the electrons that add to the metal, so the electrons on the ligands, they're going to repel the electrons in the unhybridized d orbitals of the metal atom. And then the result is that the energies of the orbitals of the d sublevel are split. So they're no longer degenerate, like we think of with an atom or an ion. Once the ligand is bound to it, those orbitals are no longer degenerate. They're not the same energy. They then split to have different energy levels. And the difference in energy levels depends on the complex and the kinds of ligands. So the crystal field splitting energy, and the strong field splitting and the weak field splitting. So let's just think about a metal ion. So the metal ion has the d orbitals, and all those d orbitals are degenerate. But now, as the initial ligands start to approach, so those ligands have these electrons, and it creates this negative charge around it. So at first, when they're far away, it's kind of a spherical effect, or it feels a spherical effect. So the electrons in the d orbitals feel the electrons from the metal, and so they become destabilized. So you get this initial electrostatic repulsion between the negative ligands and the d electron orbitals of the metal. So that results in an increase in energy because you have this repulsion of the electrons on the metal and the electrons on the ligand. Now, the ligands, as they start closing in on the metal ion, if you have really good overlap of the ligand with the d orbital, you have more repulsion. So those orbitals are going to go even more up in energy. But if the ligand comes in between the orbital that contains the electron, it's going to miss it, so it's not going to have as much overlap. So those are actually going to get stabilized. So this is where we get this loss of degeneracy of the d orbitals, meaning that they are no longer the same energy. They actually split. But overall, the energy is the same. So these three ligands go down by 2/5, and these two ligands go up by 3/5. So the overall energy is the same. And the orbitals oriented along the bonds increase in energy. So the more overlap, the more repulsions. So the more energy it's going, the more destabilized it's going to be. But orbitals oriented between the bonds, this is going to be a decrease in energy. But there's also the electrostatic attraction that results in a net decrease in energy. So this is where we typically have the metal ion, which is typically positively charged, and then the ligand, which has electron density to donate to it. That's where you get this electrostatic attraction. So overall, for the complex to be formed, these are lower in energy than where we initially started. So this is the driving force for the complex formation. The overall result is lower in energy. But remember, for a metal ion, so whatever metal ion this is, the d orbitals are completely degenerate. They're all the same energy level. So this is how we do the electron configuration for a metal ion. But once you start adding ligands to that metal, so let's say we have a metal complex with 6 ligands around it. Once you put the ligands around it, then the orbitals are no longer degenerate. They start to split. And we see this arrangement of 2 higher and 3 lower for octahedral complexes. But the splitting pattern will differ depending upon the geometry that's created by the ligands. So for an octahedral complex, this is how the ligands split. The dz squared and the x squared minus y squared actually have the most overlap with the ligands. So notice here we got the lobes here for the d orbitals. For the dz squared orbitals, you've also got the donut in the same plane as the ligands, so you get some overlap there. So the more overlap, the higher in energy. Excuse me. So the more overlap, the higher in energy that the orbitals go. And notice also with the dx squared minus y squared, you got a lot of overlap with the ligands, so this one also goes up higher in energy. But for the dxy, the yz, and the xz, notice that, so here's the lobes for the dxy. Notice that these are in between. So the ligand here are in between these lobes of the ligand. So because they're just missing it, these actually get stabilized more than the other, the dz squared and the x squared minus y squared. And if you're having trouble visualizing, this ligand is actually on top and this one is below, and it's not overlapping with the d orbital. And these pictures look all the same, it's just moved in different positions. So each one of these, the ligand is lying in between these ligands here. Whoops, I shouldn't have circled that one. Oh yes, I should have. So you'll have to know this splitting arrangement for the octahedral complex, that 2 go on top and 3 go on the bottom. So now that we know that once a ligand binds to a metal, those orbitals are no longer degenerate, we can start to explain the different colors that we see for the transition metal complexes and why they have different magnetic properties. Now, if you'll think back to some of the things that you've learned in lab, the observed color is the complementary color of the one that's absorbed. So this is the color, this purple color is that for the titanium hexaaqua complex. So there are 6 water molecules bound to this titanium 3+ ion. And if we were to take the visible spectrum of this complex, we'll see that it absorbs strongly at 498 nanometers. And 498 nanometers is in this blue-green region of the spectrum. So it's absorbing strongly here. And it even extends out into the green and the yellow spectrum. So it extends out all the way out to here. So this is the color, these are the colors that it's absorbing strongly. And because that's the light that it's absorbing strongly, it's the rest of these colors that are reflected back. So we see the complementary color of what's absorbed. So something that's absorbing violet light at 410 appears bright yellow. Something that's absorbing 500, it's absorbing the blue-green, it appears red. And you can kind of use this as a guideline. Now, when we do this by hand, we kind of do a rough estimation. So we create our color wheel. So the red, the red and the blue, red and blue make purple, and our yellow is our other primary color. So yellow and blue make green, and red and yellow make orange. Now you'll notice from this visible spectrum, notice that each of these regions where we like to see the green, like here's the green region, that's a bit smaller than, say, the red. Look how big this red region is, because we can't, our eyes don't always differentiate between these shades very well. So this approximation where we use the color wheel, that something that appears yellow is absorbing purple, this is a first approximation. I mean, it's usually a good approximation. So this is the level that we're going to be working with. But just know that maybe, let's see if we can try to find something on our color chart here. Maybe something that you predict using this doesn't appear exactly the color that you see, but this is a good approximation of the color that you should see based on what's absorbed. So again, we're looking at the visible spectrum of the titanium complex, this hexaaqua species, and it consists of a single transition at about 500 nanometers. So this is where that maximum is. But what's happening in this complex, remember we said that the d orbitals are split into 3 orbitals down low and 2 up high? Well, there's one valence electron. So if I look at my titanium, and this is a titanium 3+, and I do my electron configuration, so this would be argon 4s2 3d2, but that's for a neutral complex. We got to remove 3 electrons, so I remove the s and I remove one of the d. So the electron configuration for a titanium ion is argon 3d1, so there's 1 electron in the d orbitals. But once we put ligands around it, those orbitals split like this, so there's 1 electron in the d orbitals. Well, when we shine visible light, and remember visible light is a combination of different colors of light, it's going to absorb most strongly the light with a wavelength of 500 nanometers. And when it absorbs that light, that electron gets promoted to a higher energy level. So that's that light that's absorbed causes the excitation. The rest of the light is reflected back. And then eventually that electron goes from its excited state back to its ground state, so the more stable configuration as the electron then lowers down, and that energy is released in the form of thermal vibrations. So the energy difference between these d orbitals, this corresponds to the amount of energy of the light that causes the excitation. So the light has to be energetic enough to cause the electron to be promoted. And if you'll remember that energy is equal to, the energy of a photon is equal to hc over lambda. So the shorter the wavelength, the more energetic the electron. But the energy difference between these corresponds to the wavelength of light that's being absorbed. And so the energy difference between this is 20,300 wave numbers. Now a D9 is another example of a simple case for looking at how the electron gets excited and causes color change. So the titanium case was a D1, so there's only one electron in there. The D9 means there are 9 electrons. So in this case you have one vacant spot for one of these other electrons to move and get excited. So this is how we explain the color based on crystal field theory. So the colors of complex ions are due to the electronic transitions between the split d sublevel orbitals, so the electron being excited into them. And the wavelength of maximum absorbance can be used to determine the size of the energy gap between those electrons that are split. So let's say we have a big split versus a small split. So if I have a big split here, I'm going to require more energy, which means a smaller wavelength of light. Whereas here, where the energy gap isn't as big, I don't need as much energy, so I can use a longer wavelength of light. And if you'll remember from our spectrum for Roy G. Biv, red light has the longest, and then purple is the most energetic, and it has the shortest wavelengths. Now the strength of the crystal field depends in a large part on the ligands that are bound to the metal. And there are things that we call strong field ligands. Strong field ligands include things like cyanide, the nitrite, the ethylene diamine, and the amine. Weak field ligands include water, hydroxide, and the halogens. Weak field ligands weakly split the d orbitals. So if they're weakly split, you don't have them split as much. Strong field ligands split the orbitals a lot more. So cyanide would cause a bigger difference between the energy levels. Something like a halogen like chlorine would cause a smaller difference. So here we have a series of cobalt-3 complexes. These all have 5 electrons in the d orbitals, and they all have octahedral geometry. But what's different are the types of ligands that are bound to them. In A, there's cyanide. In B, there's the nitrite. In C, there's phenanthroline. D is ethylene diamine. E is ammonia. F is glycine. G is water. H is the oxalato ligand. And I is a carbonate. So with a cyanide, the splitting is really far apart. So 1, 2, 3, 4, 5. So the splitting is so far apart that this wavelength of light that's actually being absorbed is more in the ultraviolet region. So it's moved out of the visible spectrum. So you have to have a lot of energy to cause one of those electrons to be promoted. But as we move over to, let's say, D, where we have the ethylene diamine, these ligands aren't split quite as much. So if we see the yellow color, we would expect it to be absorbing in the purple region of the spectrum. So purple still is a high energy form of light to cause that excitation of the electrons from the lower level to the upper level. And let's go all the way down to the carbonate. So this is a weaker field ligand, so the orbitals aren't split nearly as much. So 1, 2, 3, 4, 5. So as one of those electrons gets promoted up, it doesn't require as much light, or it doesn't require as much energy. So notice that this appears green. We would expect it to be absorbing red light, and red light is a longer wavelength and a less energetic form of light. So you can kind of do that analysis as you look through this, but this is where we created the spectrochemical series, where the strong field ligands like cyanide split things the most, split the d orbitals the most, and the halogens are weak field ligands and they split things the least. And on your cover sheet for your exams, you'll actually be given the spectrochemical series, and they'll be ranked for you so that you can see. But the greater the splitting is caused by stronger field ligands. Okay, so we were looking at how the ligands split and how much they split, and we said that the type of ligands will affect how much they're split. So that's based on the spectrochemical series, where strong field ligands such as cyanide and the ethylene diamine ammonia split it the most, and weak field ligands split the d orbitals the least. So the type of ligands will affect how much the orbitals are split. Also, the charge of the metal ion will affect how much it's split. So let's say we have an iron 2 plus versus an iron versus an iron 3 plus. The iron 3 plus will split the orbitals more than an iron 2 plus will have the orbitals split. It also depends on the type of metal. So second and third row transition metals split the orbitals, the d orbitals, more than the first row. So the further down on the periodic table you go, the more the d orbitals are split, so the bigger energy gap. And the splitting also the amount of splitting is also determined by the number of ligands. So a tetrahedral complex only has 4 ligands. And the fewer the ligands, the smaller the splitting. So for tetrahedral complexes, because there's only 4 ligands, the splitting is always not that much. So tetrahedral complexes are always going to be high spin because the splitting is always very small. But for an octahedral case, you may have either high spin or low spin. So you may have either large or small splitting. But the more ligands you have, the greater the splitting it's going to be. Now we said that the electron configuration of the metal ion will split the d orbitals, and that splitting depends on the strength of the crystal field. The fourth and the fifth electrons will go into higher energy orbitals, so the dx squared minus y squared and the dz squared, if the field is weak and the energy gap is small. This leads to unpaired electrons in a paramagnetic complex. The 4th through 6th electrons will pair the electrons in the dxy, the xz, and the yz if the field is strong and the energy gap is large. This leads to paired electrons in a diamagnetic complex. So this may be a bit confusing. What does this actually mean? So let's look at this in a little bit different form. Here we have an iron cyanide complex and an iron water complex. So the water, remember, this is a weak field ligand, so weak field ligands don't split the orbitals as much. The cyanide is a strong field ligand, so we have a larger splitting of the d orbitals. So as I fill in the electrons, this iron has an oxidation state of +2. Because each of the cyanide ligands is a -1, the overall charge is 4-, so the iron has to be a +2. So if I do the electron configuration for this, I know that the iron 2+ has 6 electrons in the d orbitals. So as I start filling in my 6 electrons, I still use my Hund's rule and my Aufbau principle. So I'm putting in 1, 2, 3 electrons. Now I've still got 3 more electrons to put in. My choice for my 4th electron, I could either put it down here low or here up high. But there's a big energy gap between these two, and I'd rather put it down low, even though it costs me some energy to pair up the electrons. It costs less energy to pair them up than it would be to put it up high. So I'm going to put them all down low. So in this case, I have a diamagnetic compound because all of my electrons are paired up. So that's what the di means. It means you've got 2. So each electron has a pair, so they're completely paired up. So this is a diamagnetic complex. And this is also termed a low spin complex. So the spin is the number of unpaired electrons. So since this has a spin value of 0, it has a low spin value because the spin value is 1/2 times the number of unpaired electrons. Now let's compare this water complex. So each water molecule is neutral, and the overall charge is 2 plus. So this means that the iron also has an oxidation state of 2 plus. So again, iron 2 plus is a D6. So there's 6 electrons. So I fill in my electrons. I start again like I did before. 1, 2, 3 electrons down low because I followed my Aufbau and my Hund's rule. Now with my 4th electron, I can either pair it up or go up high. But pairing the electrons costs some energy, but also putting it up high costs some energy. But when I compare this, for this paramagnetic high spin complex, and because the splitting small, it's easier to promote that electron, so the 4th, 5th electron. Now I've still got one more to put in, so I'm going to go back and fill in my other ones completely. So this is what we call a high spin complex, because if I count the number of unpaired electrons, I have 1, 2, 3, 4. So my spin value is 1/2 times 4, so the spin value is 2. So compared to the S equals 0, this is a high spin value because the number is higher. Now, because we put the electrons up high and we have unpaired electrons here, we have a paramagnetic compound. So anytime you have at least one unpaired electron, the compound is paramagnetic. And paramagnetic, so you've got parallel spin, so you have at least one unpaired electron. Now experimentally, I can distinguish between these two types of complexes because diamagnetic compounds behave differently in a magnetic field than paramagnetic compounds. Diamagnetic compounds are very, very weakly repelled by a magnetic field, and paramagnetic compounds are slightly attracted to a magnetic field. So just by putting them into a magnetic field and seeing how they respond, I can figure out how many unpaired electrons that they have. And so I can distinguish between a low spin versus a high spin configuration. Now if I'm looking at these configurations, only the D4, 5, 6, or 7 can have high or low spin. Because if I have, let's say I have 1, 2, or 3 electrons, 1, I can only fill it in one way. If I have 2, I can only fill it in one way. If I have 3, I can only fill it in one way. If I do my 4th electron, I have a choice, either go high spin or low spin. So 4th, 5th, and 6th, and 7th, I have choices where to put the electrons. So 1, 2, 3, 4, 5, 6, 7. So for D8 complexes, there's only one way to do it. So the 8, and then the 9, and then the 10. There's only one way I can arrange those electrons. So I don't have to distinguish between high spin or low spin for D8, 9, or 10 complexes. So again, like I said, I can determine the magnetism of the compound to figure out how the electrons are arranged. And I can use what's called a magnetic susceptibility balance. So what it does is it figures out how susceptible your sample is to a magnetic field. And in this, so in the top here, this is where you insert your sample into a little glass tube. You insert it in here. So there's your sample here, and it puts it into a magnetic field. And depending upon how it responds, so if it's diamagnetic, it'll be weakly repelled by the magnetic field, and if it's paramagnetic, it'll be attracted to the magnetic field, so it will affect the observed mass of it. So the mass really doesn't change, but it kind of feels like the mass changes because it's being influenced by the magnets. So for a diamagnetic complex, all the electrons are completely paired up. So you have no unpaired electrons. And they're going to be weakly repelled by a magnetic field. But paramagnetic complexes have at least one unpaired electron. And each one of these arrows represents a complex with unpaired electrons. and they're all arranged in different orientations, a point in different directions. But if I were to put this into a magnetic field, the arrows or the electrons would align with the magnetic field. And when they do, they're attracted to the magnetic field. So paramagnetic compounds are attracted to a magnetic field. Now there's another type of magnetism called ferromagnetic. So this is things like your refrigerator magnets. So things that you think of as being magnets, those are ferromagnetic materials. And ferromagnetic materials have regions. So this is a sample, and in the different regions you have the different atoms with the unpaired electrons, and within the region they all line up together. And if I were to put this into a magnetic field, those unpaired electrons will also align with the magnetic field. But ferromagnetic compounds are very strongly attracted to a magnetic field because you have these magnetic domains. And if you're at a certain temperature, when you remove the magnetic field, they'll all stay aligned together, which makes them magnetic and stick to like your refrigerator door. Now again, remember we said that if you put the ligands in different geometries, you get different splitting. So this is the splitting for a tetrahedral complex. So the x squared minus y squared and the dz squared are lower in energy than the xy, the yz, and the xz. Because the ligand approach interacts more strongly with the planar orbitals in the tetrahedral geometries, their energies are raised, and most of the tetrahedral complexes are going to be high spin. So all the instances that we're going to be working with, if it's tetrahedral, we're going to treat it like a high spin complex. So let's say we had a D5. So 1, 2, 3, 4, 5. Now, if we're looking at a square planar complex, so all the ligands are in the equatorial plane, so you have a lot of good overlap with the x squared minus y squared ligand, or the x squared minus y squared orbital with the ligand. So this is going to be highest in energy. And then the xy, so it lies in the same plane as the ligand, so that's going to be destabilized, but not as much as the x squared minus y squared. We have direct overlap. Then you have the dz squared because you have a lot of electron density above and below the plane. And then the most stabilized will be the xz and the yz. Now if you needed the splitting pattern for a square planar complex to work a problem, we would give it to you. You do not have to memorize this one. And we typically see square planar complexes with D8 metal. So if you do the electron configuration for your metal and you see it's a D8 metal and there are 4 ligands attached to it, treat it like it's square planar. And the D8 complexes, most of them are going to be low spin. So let's say it's 8 electrons, we're going to treat it as low spin. So 1, 2, 3, 4, 5, 6, 7, 8. Because there's a big energy gap in between these orbitals. So the electron doesn't get promoted. Now here's just some application of coordination compounds. For example, you can use coordination chemistry to extract metals from ores. So silver and gold are removed from minerals as cyanide complexes, and then you can reduce it back to the elemental forms for silver and gold. Nickel is actually extracted as nickel tetracarbonyl, and nickel tetracarbonyl is actually a very, very toxic compound. It's probably one of the most toxic coordination compounds known because it's actually in the gaseous form. But by adding carbon monoxide to the nickel in the ore, it extracts it out as this gas, and then you can convert the gas back into the elemental or the reduced metal form of the nickel to get the metal out. You can also use the ligands as chelating agents in case you're ever exposed to heavy metal poisoning. So for example, if you're exposed to lead and somehow you've ingested it or you've been exposed to lead and you go to the doctor, you may get a chelation therapy treatment, and the chelating ligand that they may give you is EDTA. You can also use coordination compounds for chemical analysis. So think about when in the lab you made the iron thiocyanate complex when you were doing the Intro to Equilibrium lab. So the iron complex, or the iron metal, so the iron ion was either colorless or lightly orange color, and the thiocyanide ion was colorless. And when you combine the two, you got this really dark blood red orange color. If you add thiocyanide to cobalt, you get a blue color. So this is actually used in the Scott test for detecting cocaine. So the Scott reagent test, this is a test little packet that they actually sell to law enforcement. And inside they've got some vials of some reagents. So basically a little Ziploc baggie that the officer then puts what he thinks may be cocaine into the baggie. He seals it up and he breaks the ampules. So after breaking the first ampule, it'll turn blue. And then you add the second ampule, or you break the second ampule and turn pink. And then you break the third ampule. And if there's cocaine present in it, you'll actually see a blue solid precipitate out of a pink solution. So that's how you can do a quick field test for the presence of cocaine. And there's some really cool acid-base chemistry going on here. So this is a structure of cocaine, and this nitrogen here has lone pairs. And after our sequence with acid-base chemistry, we should think that that nitrogen there should act like a weak base. And if we add an acid, so we add a proton, it will protonate that nitrogen there to make this cation form. So the proton from the acid protonates the nitrogen to form this cation. And then what happens, part of that Scott reagent is this cobalt thiocyanide complex, and it's pink. And when you protonate and you add the cocaine, so you add the acid and you add the cocaine, and it protonates the cocaine, and then when that reacts with the pink cobalt thiocyanide complex, it produces this new complex ion. And here the cobalt goes from an octahedral complex to a tetrahedral complex, and this complex here has very poor solubility. So going from the octahedral pink form gets converted to the tetrahedral blue form, and the solubility is very poor. It precipitates out. And this is how you actually detect that cocaine is present. So some other applications of coordination compounds, we talked about how the compounds are very colorful, so they can be used in inks and blueprinting and cosmetics and paints. There are also a lot of biomolecules. For example, the porphyrin ring. So the porphyrin ring actually adds a lot of stability. So having the ligand in the form of the ring here makes it for a very stable complex, because in this case all the metal has to do is fit inside the hole, and the ligand is already prearranged to fit around the metal. So think about this analogy. Let's say you've got two bracelets to put on. So you've got your hand here and you want to put on a bracelet, but one of your bracelets, you've got the hook on one side and you've got to hook it to the other end. Or you've got a bracelet where it's just a bangle. So the bracelet that you have to wrap around your wrist is a bit harder to put on than the bangle, because all you got to do is slip your hand through the hole here to put the bracelet on. Well, it's kind of the same way with ligands. If the ligand, so let's say we have this ligand here, we've got these nitrogen donors. For this ligand, I've got to wrap it around the metal. But if the nitrogens are already arranged in a circle like they are in the porphyrin, all the metal's got to do is fit inside the hole. So this is called the macrocyclic effect. And if the ligand uses a macrocyclic effect, it will have a larger formation constant, and the complex is much more likely to form. So this is why biology uses this porphyrin molecule, this porphyrin ligand, because it forms a very stable metal complex. So we see porphyrin rings in things such as cytochrome C, also in hemoglobin. So this is a hemoglobin protein. There are 2 alpha and 2 beta subunits. So we have a subunit here and a subunit here. And you can see the porphyrin ring, the heme centers in each part of the protein there. So you have the porphyrin ligand around it, and you have the iron that sits in the center. And it's the iron that carries the oxygen. So the oxygen binds to the iron and it's transported through the blood. And porphyrin rings are also used in chlorophyll. And here's an example of a chlorophyll. And the center of the rings, the metal that's used in it, is actually a magnesium. So in the antenna, for the receptors that receive the light for photosynthesis, it uses these chlorophyll molecules with the magnesium in the center of a porphyrin ring. Another coordination compound that's very important is carbonic anhydrase. This is actually a zinc metal center, and it's got 3 histidine ligands around it. And this enzyme is actually responsible for buffering your pH of your blood, keeping it at a certain pH level. So it catalyzes a reaction between water and CO2 to form the carbonate. So carbonic anhydrases are actually one of the fastest known enzymes, and enzymes are just biological catalysts, so it catalyzes that conversion between the reaction of water and CO2. So it helps control the pH of your blood. It also, carbonic anhydrases are also very useful in other biological systems. So for example, it helps control also helps control the pressure in your eye. And another example of a coordination compound is cisplatin. This is actually an anti-cancer drug. So if you think back to like Lance Armstrong in his fight with testicular cancer, the drug that they gave him to treat his testicular cancer was cisplatin. So this is a platinum complex. So you have a platinum molecule with 2 amines there. So it's a platinum with 2 NH3s attached to it. And you've got the chloroligands. Now once that enters the body, those chlorines are then replaced with water molecules. And then those water molecules are still pretty labile, and so you lose those water molecules, and the platinum will then attach to the bases in the DNA. So here you can see the platinum bind to the DNA, and it causes this kink or folding in the DNA. And when it does, the DNA can't be repaired by the repair processes. And so what happens is that signals apoptosis, so it triggers cell death. So this is how the cisplatin drug works. So it causes a kink in the DNA, and the DNA can't be repaired, so the cell then gives a signal to kill itself, to go through apoptosis, because it can't repair the DNA of the kink. So it kills the cancer cell. So that ends our chapter 24, where we're looking at transition metals. So remember, if you've got questions, be sure to bring them to class, and we'll work through some problems to make sure you understand a lot of these concepts.